Welcome back to Decouple, a special recording from the Canadian Nuclear Association Conference here in Ottawa, Canada. And today I'm joined by my good friend, Douglas Boreham, veteran of the podcast, radiation biologist. You have a lot of qualifications that I'm going to stumble over, Doug. So
don't worry about it.
Why don't you do the famous self-introduction? Just give us a tidbit more than I've...
All right, so I'm a radiation biologist. I worked 10 years at Atomic Energy of Canada in Chalk River, then went 12 years at McMaster University in their medical physics department. And now I am the division head of the medical school, or now the Northern Ontario School of Medicine University. Division head. On the side, I also worked 12 years at Bruce Power.
Okay.
Excellent. So you add that up, it's a lot of years.
That's a fair number of years. I'm doing some calculations in my head right now.
Yeah.
Douglas, I mean, I'm really lucky. I have a lot of excellent guests on the podcast who then sort of become sort of special advisors that I can reach out to with questions. And we had a really interesting interaction recently because my dad is actually being treated for prostate cancer with the medical isotope lutetium-177, which Bruce Power is working on being a major supplier for.
Right.
But I looked at his hospital discharge instructions, and I had a sense of how insane they were, but I'll just share some details. He was supposed to move his bed 3 meters from the nearest wall.
We had a big house.
Yeah, exactly. I mean, who can do that? No public transport. Couldn't sleep in the same bed as my mother for quite a while. Had to flush the toilet 3 times after every use, and then do a full swab down of the bathroom. I think after week 1.
Yeah.
So I remember reaching out to you and saying, Doug, this sounds insane to me, but it's great to I'm not a radiation biologist, so it was it's just a pleasure having you in this network of
so we can figure out
experts.
That it's a bit of an overreaction. We looked into it, and I think some of these regulations that we have, or guidelines that we have for medical isotope uses in the future, are going to have to change a little bit, because it is not only is it inconvenient for the patient, but it's quality of life too, right? And you're getting treated for a disease that's really bad disease, and now you have to go through all this other stuff when really you don't have to. In fact, most of that stuff is out of the body from a biological half-life within a couple hours. Yeah. And then the other stuff, because of the half-life it has, it decays away in a very short period of time. And the dose rate that you're giving off to someone beside you is the almost identical dose rate that you get sitting on an airplane at 30,000 feet, which is 4 microsieverts an hour, which is biologically a non-starter.
So, you take a cancer patient who's got enough on their plate dealing with that diagnosis and who needs the support of, not just their community, but their family, and you say you got to basically sit in a room all by yourself 3 meters from the nearest wall. And that causes real harm, and I don't think, I think we have a real hard time with understanding risk in our society and weighing risk and benefit. Anyway, it was very useful to
call me up.
To go from about 95% certainty that this was insane to
to get some facts
close as
on
100% as you can get. So, Douglas, one of the reasons that our listeners really enjoyed your episode is you bring a lot of, for our science nerd audience, you bring a lot of really cool stories.
Yep. That's why I do the science. I only do if it's going to be fun, right? I mean, what's the point if it's not going to be fun?
Awesome. So we talked a little bit in the podcast about Snow Lab. Right. And I think we're going to talk with one of your colleagues a little bit about the research that's being done on radon and, basically no radiation environments. Yep. But you were just telling me about the NASA project sending yeast to space. So let's, for our science nerd audience, let's
start with that.
Chat a bit about that.
You heard me just mention a second ago about the radiation that people get flying. Well, that's cosmic radiation, right? There's a lot of cosmic radiation out there, especially when you get outside the atmosphere, above and beyond low Earth orbit where the space station is. We're sending people to the moon next year, or 2024, and then on to Mars. And we have many challenges as a human in space, but one of them is cosmic radiation, and that's where we come into it. We're collaborating with NASA, and their punchline is, NASA sending yeast into space where no yeast has gone before. Right. And it's very interesting because yeast, from the perspective of research, are a perfect astronaut. Because right now we don't have the ability to send a living thing deep into space, and we're talking 40 million kilometers out into space, and be able to monitor it and know it's alive, and then assess the effects of this cosmic radiation that's out there. And the astronauts, even at low Earth orbit, or when they went to the Apollo mission, they get hit with a lot of cosmic radiation, and to the extent where they get flashes of light in their eyes from heavily charged particles going through their head, their retina, and back out their head again. So there's going to be biological effects of this kind of stuff.
Right.
So by sending these yeast out into space, and the reason I say they're perfect astronauts is because they don't need to drink, they don't need water, they don't need to breathe. That's an important thing. They don't really care about the temperature. And so they're very low maintenance. So they don't eat, they don't breathe, they don't need water, and they don't care about what the temperature is. So now they're off, they go into space, and about every couple of weeks, a little robot on this yeast spaceship wakes a couple of them up.
Right.
So they're in a state of what we call anhydrobiosis, which is life without water.
Right.
And when you rehydrate them, they immediately come to life. When you make your pizza or your beer, you put in freeze-dried yeast, poof, they wake up.
Yeah.
They're a eukaryotic cell, which is a cell like we're made up of 100 trillion eukaryotic cells.
Right.
These are individual eukaryotic cells. They're in a desiccated state, and NASA's very interested in that because the desiccated state could almost be like a state of hibernation. And part of what the research that we're doing is we're trying to figure out how we can put humans into hibernation.
Wow.
To go to Mars. It'd make the trip a lot easier.
How many days is it currently to arrive there?
6 months.
When the planets line up correctly and you can slingshot someone there?
6 months.
Wow.
Yeah, and then 6 months to get back. And then 6 months you're going to hang out there. So,
Jesus.
yeah, it's going to be a stressful thing. So
and no hanky-panky in space during
apparently
that
not.
time.
That's what they're suggesting. That's not a good idea.
Right.
I'm sure they've tried it already on the International Space Station, but I'm not going to go. I don't know. I just made that up.
Right.
Hypothetically. But yeah, so these yeast are going into space, are getting irradiated with this cosmic radiation, and when the robot wakes them up, we can get a determination of how well they're repairing themselves as they go deeper and deeper into space.
Right.
And we also have two strains of yeast. One that has the inability to fix what we call DNA double-strand breaks, which are the critical lesions when it comes to the effects of radiation.
Right.
I mean, you want to have a lot of double-strand breaks in tumor tissue when you irradiate it. You want to kill them, and that's how you kill them, by making double-strand breaks in DNA. The yeast that don't have that ability to repair are going to be impacted much more by the cosmic radiation.
So they've been genetically engineered to lack that repair mechanism.
One gene's been knocked out. It's a gene that has a repair system called homologous recombination. And that allows them to fix double-strand breaks and not make mistakes. So in other words, not mutate.
Right.
One yeast lacks that ability. The other yeast is what we call the wild-type yeast, and they're going off into space. And so what we're doing is we're looking at how these things are going to behave to different types of radiation. And so we have the Snow Lab, which you'll hear about, that has no cosmic radiation and no other types of radiation. And we're going to see how those yeast survive under those circumstances, and how they're surviving in deep space, and that's the way to compare what biological mechanisms are actually at play by keeping eukaryotic cells alive in space.
Right.
And then there's going to be a number of countermeasures that we're going to try and come up with to protect the eukaryotic cell from this cosmic radiation.
Right.
We've taken these yeast in this desiccated state to irradiation facilities that can give very high doses of radiation. Now, for example, if you're getting cancer treatment, you get about 2 gray per fraction, right? 2 gray a day.
And as a whole body dose, that's enough to
a whole body dose of 2 gray,
radiation
that would be
syndrome.
A bit high. Some people would definitely notice a 2 gray whole body dose, but a 2 gray dose to the tumor itself,
exactly, right.
That's where you get the effect. And as you know, you don't put the 2 gray into the tumor all at once. You put it in at different angles and different different beams, and then that way the normal tissue around it only gets a little bit of radiation, but it all builds up in the tumor to give you your 2 gray.
Exactly. Yeah.
That 2 gray then causes roughly 80 or 100 double-strand breaks in every cell, and that can kill the cells. Not all of them, but they'll repair, and then you do it again, That's why you go every day to get this radiation therapy.
Right.
These desiccated yeast, we've given them already up to 2,000 gray. So 1,000 times more than what you get in a radiation therapy treatment. And about 30% of them survive, which is amazing when you think about the dose. So part of what we're doing with this is an extension that we can actually use this yeast model system to look at how double-strand breaks are formed and how cells repair. And one of the newest radiation therapy treatments that we're coming up with is called flash radiation.
Yeah.
And that's instead of giving a 2 gray dose of radiation to a tumor roughly over 30 seconds or a minute, this happens in a millisecond.
Wow.
So they can deliver doses in what's called flash radiation, so 100 gray a second.
Jesus.
That's a lot of dose. So it's a flash, right? And the interesting thing about it is that if they give flash radiation to a tumor, it looks like the tumor cells are affected, like the yeast that can't repair itself, but the normal tissue isn't. So it protects normal tissue, flash radiation.
Wow, okay.
We're going to take the yeast system and we're going to do flash radiation on them, and that'll help us understand how giving a dose of, it took us 24 hours to give a dose of 2,000 gray, and we're going to give a dose of 2,000 gray in literally less than a few minutes.
So huge applications back here on Earth. But you mentioned a couple statistics around these double-stranded DNA breaks, and one of the big things that the takeaway I think from our podcast was just at the dawn of genetics and understanding the DNA double helix, et cetera, we didn't have that appreciation of the repair mechanism. So I just wanted to, I think that's a great thing to demystify. In terms of single strand and double strand breaks, how often are those just happening spontaneously in our cells as a result of, oxidative metabolism?
Oxidative metabolism. So I guess the example I would use is if you get a CT scan, you get what we call 10 milligray of dose. That's roughly equivalent to 10 photons through each cell in your body.
Yeah.
If you're sitting, waiting in the waiting room, breathing oxygen for an hour, getting oxidative metabolism damage, there's 10,000 times more damage in that 1 hour from breathing oxygen there is from the actual photons caused by the CT scan. So people don't appreciate the whole idea of oxidative stress and oxidative metabolism and how that damages their DNA. I mean, single strand breaks, if you think about it, we have two strands of DNA and they're complementary to each other. If you get a single strand break in one, a repair system can fix it easy because the template is the duplicate copy, right? Yeah. So you have a backup copy, you just patch the hole and you're good to go. When you break two strands, now where do you get the information? Well, we just so happen to have two sets of chromosomes, right? We have two number ones, two number twos. So if you break a double strand break in chromosome number 1 here, you find the homologous chromosome and you can do what we call homologous recombination. So you actually have backup information on your second copy.
Wow.
And that
backup hard drive.
Backup hard drive. It's your flash drive. You got two of them, right? So this one doesn't work. You copy the information over, and now it's repaired in the same way it was. What happens is sometimes there's mistakes being made. And that usually happens from what we call non-homologous end rejoining. And that's when the cell just sticks it together and doesn't put in the right bases. That causes a problem after that. So other than that, it's...
But I've heard, I think, in the average cell, there's 10,000 single-strand breaks a day, something like that?
Oh, it's more than that. I think it's an hour. It's I have to get the number on that. But
I'm ambushing you with that.
It's 10,000 DNA alterations per hour in a cell. Wow. Not necessarily single strand breaks, but so like a 1 gray dose of radiation in one cell will probably cause 2,000 to 3,000 single strand breaks and somewhere around 40 to 50 double strand breaks.
Right.
A single gray.
Right.
And cells can repair that. They can handle that and they can manage it. So you mentioned DNA repair, but we also have mechanisms in our cells that will say, okay, the repair system somehow failed and the DNA is not right. So what the cell does then is turns on genes that are called tumor suppressor genes, which causes cells to self-destruct.
Seppuku, as a Japanese say.
Exactly. And you know what? A dead cell can't become a cancer cell.
Right.
And we're roughly having in our body 3 million cells commit suicide every minute. Yeah, that's to keep the genomic stability, and we replace 3 million cells every minute.
Right.
And that's what keeps us at balance and keeps the cells.
It's funny you mentioned that CT scan, because I think there's a strength that I have as a physician, and you have as someone who works at a medical school, being able to compare radiation doses to everyday things that people experience. And we have this background radiation dose, and the component that's artificial is about 15%, and 14% of that is medical. But I was talking to Dr. Geraldine Thomas of the Chernobyl Tissue Bank, and she was saying, the 6 million people around Chernobyl that were most affected got the equivalent radiation dose of a single whole body CT scan, but over a 25-year period.
Right.
So it gives you a little bit of a sense, because we really are so terrified of radiation, and it really is affecting the choices that we're making and responding to climate change, or
Exactly. And I think there's still a lot of people that believe, and we were talking today about theology and math. I don't know if you remember that talk this morning. There's a lot of people that believe that radiation doses add up. You get one today, you get one tomorrow, you get one next week. These add up over your lifetime, and your lifetime risk then goes up. Our research, and a number of researchers in my area, we don't accept that hypothesis or that idea.
Right.
Because if you think about all the things that damage our body, including alcohol, if it was to add up, we'd all be dead.
Right. And working out would be a really bad idea.
Bad idea, yeah. So you see, it's a really interesting analogy that you can compare things. And like I said to you before, when you spread things out over time, our cells have more time to repair. Right. Our cells have a easier time of fixing. So if you get a double strand break now and you fix it, and you get one another hour from now, you fix it. If you get like 40 of them all at once, and you're going to probably die. The cell's going to die.
So how long is it going to take for the radiation protection establishment to kind of catch up with the science?
Well, I started doing this stuff in 1986. Actually, I started my PhD the year Chernobyl went up in August of '86. And in those days, the whole idea was, when are we going to actually figure out that there's no such thing as a linear response to radiation, and there's no such thing as additive risk, and there's no such thing as a collective dose. At least the collective dose thing is gone away, right?
Right.
But
from Unskier, I mean, not from Greenpeace, but
no,
from,
it just went away because the scientists finally figured out that
this was a step too far. This was too.
You just can't add up little doses of a big population and calculate risk to one person from all those doses.
Right.
That's a collective dose thing.
And that's where you get these big numbers when Greenpeace says 200,000 people will die from Chernobyl, or the Torch report in Europe, 3 very ideological scientists.
Well, and you use the model, I think they were predicting some 4,000 increased cancer risk deaths in the Chernobyl incident, and I don't think there was any increase. So, that and
except for thyroid cancer, obviously.
Yeah, the thyroid cancer has always been a mystery to some people too, because in terms of what we call latency period, from when the time you get exposed to a carcinogen and the time when the certain tumors appear, is a latency period. It's the time it takes for the cells to go through the multi-step processes and become a cancer cell. What happened with the Chernobyl thing is that only a year after these exposures to the iodine, which we know is an increased cancer risk, especially in children, all these thyroid cancers started showing up. And some of the argument is that, well, if you go start looking for stuff, you're going to find things, even if they're occult, meaning they're not going to cause any problems, they're just a benign tumor sitting there. But they start finding all these things. So they showed a statistical increase. It's still unclear to a lot of people, was this a real phenomenon of actual iodine-induced thyroid cancer, or it was a consequence of the medical process of looking for things and finding things.
What percentage is
and what was caused.
Screening effect.
I think. But the other thing people should know or recognize is that some people are getting thyroid cancer, but it's not a lethal type of cancer. It can be treated with the actual iodine that caused it. Right. So
it's not quite homeopathy because you're not diluting it down, but you use the exact same isotope.
Same isotope that caused it. You use it to treat it with.
Yeah. All right, Doug. I mean, I could talk to you all day.
Sure.
We were supposed to talk for 5 minutes, but here we are about 20 minutes later.
20 minutes.
Pleasure chatting with you.
No worry.
To be repeated. So thanks again for making the time.
Cheers.
Okay.